Very little basic research has been performed in the field of drug resistance or in understanding why cancer therapies fail. And I think that's because it has conventionally been under the purview of clinicians to try new therapies, essentially by a trial and error mechanism. You have patients that present with identical looking tumors. So you have two tumors that appear exactly the same. One responds to therapy and the other doesn't. And so we want to understand what the basis of that success or failure is. One of the big challenges in cancer biology is understanding not how one gene functions or two or three, but how an entire system works. How pathways leading from a drug to a chemotherapeutic response is worked out by the thousands of genes and proteins that are in a cell. And so there's an entire field called systems biology that's focused on this question. One of the main objectives of our laboratory is actually to be able to perform high throughput genetics in model systems that allow us to essentially screen for mechanisms of drug resistance. Now, this is a kind of technology that's only evolved over the last few years, and it's evolved in places like MIT. In fact, a great deal of work in developing this technology has been done here, so we can now start looking at 10,000 or 20,000 genes at once. So we're accumulating this huge amount of data from genome sequencing efforts. And so one question is, how do you deal with all of this information? One of my graduate students, Justin Pritchard, is co-advised by me and Doug Laufenberger. The computational side, Doug knows very well, and the wet bench side, I know extremely well. And I think here we look to the highest common denominator and not the lowest. So in essence, we're getting the best of both worlds. I think the Koch Institute is fundamentally enabling. So it allows for connections between people and scientists that you couldn't find anywhere else.